Hi there, welcome to this video on working out monohybrid crosses. The specification point is listed for you here. Just to remind you, you need to be able to describe the patterns of monohybrid crossing, and we will take a look for that 3-to-1 ratio that Mendel produced using genetic diagrams, and we tend to use Punnett squares. And the 3-to-1 ratio is a probability of the outcomes of the crosses, which we're going to achieve. And just clean this up. Now, the first thing we want to do is figure out from the question you've been asked what you are crossing. So for our first one, we're going to, first example, let's identify what we have. So we have our parental phenotype, and the phenotype are the features of the organisms that we're crossing. So I want to cross two humans, one with brown eyes and one with blue. So we'll have blue eyes and brown. Now these two individuals are going to be homozygous. Homo meaning same, so their alleles will be the same. So they're both homozygous. So the first thing we identify after the parental phenotypes is the parental genotype. Now, I've got these in an order down the left-hand side of the page. You need to label them as you do your work. So the parental genotype for our homozygous parent will be little b, little b. Both alleles, we represent them as lowercase because they are recessive to brown. Brown eyes, homozygous, big B, big B. So the parent, all of their cells, their body cells, contain these two alleles. The next thing we need to identify is the gamete genotype. So the sex cells from these parents, let's say we have the blue-eyed individual is female, and when the gametes are being formed, we get one gamete which we put a circle around to show that it is completely separate, has little b, and the other one has little b. The other parent, let's say it's the father, has brown eyes, so one sperm is carrying the big B allele and the other sperm is carrying the big B allele. Next, we do our cross. So let's say this was the father at the top, and we've got mother on the left-hand side, and mother contributes eggs with little b and little b. Father contributes alleles big B and big B. So let's cross them over. Big B, little b, when fertilization occurs, and we always put the dominant allele first. So we've done our genetic diagram, and now we identify the F1 offspring genotype. So the possible children that are going to produce by these individuals, what are their genotypes going to be? Well, we've got 1, 2, 3, 4 are all big B, little b. Now we can write them all out just to be absolutely sure. I'm using commas. Now, since they're all big B, little b, we could just write it once. But thoroughness. And F1 for the record stands for first filial. Unlikely to come into contact with this, but filial refers to family. F1 phenotype, so the features of these offspring. Well, they're all big B, little b. They have a dominant allele for brown eyes and a recessive allele for blue eyes, which means this individual is brown, as does this one. Brown, brown, brown. Okay, so we have 100% brown eyes. Okay, I thought I said something about a 3-to-1 ratio. Well, check back in the next video and I'll show you what happens when we cross heterozygous individuals together. Thank you.